Chapter 6, Section 3, Templates, Enzymes, and the Replication Fork. Now, hopefully you remember from your Intro Cell and Molecular, your cell biology or genetics courses, that DNA replication for all living things is semi-conservative. Okay, it's what Watson and Crick figured out. Kind of the part of the reason, big reason why they got that whole Nobel Prize thing. Semi-conservative, that the double helix, the two strands, will open up, forming that replication bubble, and each of the strands will act as a template for the making of the new. So, the precursor for each nucleotide is a deoxynucleoside 5' triphosphate. Okay, what does that mean? Well, the A, the G's, the C's, the T nucleotides start off as a trinucleotide, 3 phosphate groups there, okay? It's not ATP, it's not GTP, as you would know it from, metabolic reactions, because this is a deoxyribonucleic acid nucleotide. It always starts from the 5' end, okay? The 5' end, meaning that there is this open 3'. The 5' end means there's a 5' phosphate sticking up there. Okay? This, right there. This is the 5' end. Right here is the 3' end. This is where the nucleotide will be added in. This is what DNA polymerase must have to build on. It adds in the incoming nucleotide by cutting right here. Cuts right there, because right here is the 1, the 2, the 3 of the trinucleotides. If you take biochemistry, you'll see this is called the alpha, the beta, the gamma. The beta and the gamma are cut off, and in the process of cutting them off, there's all that energy right there that's going to be imbued into the molecule, into the reaction, to have this bond reform right here. Here's a little video. You can go and look at the handouts and you can watch this little video that will help illustrate what we're talking about. I'm not going to sit here and play it here. You can watch it on your own. DNA polymerases catalyze, okay, this reaction. They catalyze the addition of the incoming triphosphate nucleotide into the growing polynucleotide. They add it at the open 3' end. And there are 5 DNA polymerases in E. coli. DNA polymerase 1, 2, 3, 4, 5. Notice it is numbered, numerals 1, 2, 3, 4, 5. If this was Greek letters, that would signify mammalian, eukaryotic. 1, 2, 3, 4, 5 is going to be prokaryotic. DNA polymerase 3 is the primary enzyme replicating the DNA. Why is 3 the most important one? Why isn't it 1 or any of those? Basically, they're ordered in how they found them. Hey, here's the first one we found. Well, here's another one. Oh, this will be 1, this will be 2, and so on. Turns out 3 is the one that does the most work. DNA polymerase 1 comes in, and this is the one that's going to do come to play on the when it comes into removing the RNA primers for the Okazaki fragments, which we'll talk about in a few minutes. The others, 2, 4, 5, play roles in damage repair. Here's the problem, though. Because of the way they're structured, DNA polymerases can only add to pre-existing nucleotide strands. they have to have a 3' hydroxyl to build upon. They cannot just grab onto a single-strand DNA and start making the complementary strand. So they have to have a primer, a short strand of RNA to build on. The RNA is laid down, complementary base pairing, of course, by the enzyme known as primase. Okay, primase lays down a short little strand of RNA in the double helix, which then DNA polymerase III comes and starts building upon. DNA Pol I will come in later, remove those RNA nucleotides, lay down DNA nucleotides, excuse me. Here's all the major players you're going to see in bacteria. Primase, DNA polymerase III, DNA N, gyrases, DNA A. We've talked DNA A before. Remember, this is the one that binds at the origin. Starts off the whole origin of replication thing. DNA B, C, and so on and so forth. So you have the template strand. You don't know what to do with it. Okay, not a big deal, because DNA polymerase can't bind, and if it can interact, there's nothing it can build on. So RNA primase comes and lays down a primer. Primer is going to be anywhere from 1 to maybe 2 or 3 dozen nucleotides, just long enough to be stable. And then DNA polymerase comes in and starts building upon it. DNA Pol III. Pol I comes in, removes the RNA primer, lays down DNA. So it's now all DNA, no RNA. All of this work is happening in what's referred to as a replication fork. Okay, the replication fork. Remember, this term fork used to refer to a road that splits and goes into 2 directions, a fork in the road. This is where they got that idea, replication fork. One double helix splits and becomes 2. At this replication fork, you're going to have enzymes like helicase that's unwinding. You're going to have a bunch of other proteins there, DNA A and others, that are going to bind and begin this replication process. Remember, the origin of replication is always going to be right here at ORI-C, origin C. This is a section of DNA that, well, DNA A is going to bind. DNA binds because it begins the unwinding of the double helix. Then you're going to have DNA B, the helicase, and DNA C. Some loader proteins come in, and they're going to stabilize everything there. Bring in the primase, bring in DNA polymerase, and then everything just takes off. As long as the helicase is moving forward, and then all the other enzymes will be coming along afterwards. Helicase is unwinding. Its job is to act as a zipper, unzipping the double helix. Hopefully you remember from your intracellular molecular genetics that there's a leading and a lagging strand. Leading strand, everything's in the correct orientation. Lay down one primer and go. Turns out, though, that the other strand, the non-leading strand, is in the wrong orientation. So you're going to have this lagging strand, this series of the complementary strands laid down. Each series, each short segment is going to be initiated by a new primer. DNA Pol III is going to jump primer to primer, laying down. DNA Pol I is going to come in and remove all those primers, lay down new DNA. Ligase is going to seal everything up. So you have your supercoiled chromosome here in the nucleoid. One at this fork here, one strand is in the correct orientation, lay down one primer, and then just go. Follow the helicase. As it unwinds, DNA Pol III is right behind it. moving along, laying down a new. But this, the lagging strand, is in the wrong orientation. It's backwards. So you have to lay down all these primers, and then Paul 3 fills in between a primer, jumps back, fills in, jumps back, fills in, jumps back, fills in. Paul 1 comes through, removes all the RNA primers, replaces them with DNA nucleotides. Ligase comes through, joins everything together as it's showing here. Paul 3 fills in, hits this, jumps back to another one here, fills in, jumps back. Paul 1 comes in, removes the RNA, lays down new DNA. Ligase joins everything, and once they're done with their little dance, you have a continuous new strand of DNA. You have a double helix.